Okay, let's unpack this. The headline of protein folding being solved. Well, that might be technically true in some ways, but the real work, the deep engineering challenge, the making these models faster, broader, and I think critically much more useful for things like rational drug discovery and functional annotation. That's where it gets really interesting now.
Yeah, absolutely. We've definitely moved far beyond that initial breakthrough moment. We're now deep into this phase of, well, aggressive open source specialization in structural biology AI. It's really not about if we can predict single chain structures accurately anymore.
Right.
The focus, it seems, has entirely shifted. It's about generalization, achieving these orders of magnitude speed improvements, and maybe most importantly, predicting biological function and that tricky binding affinity.
So our mission today is a deep dive into exactly that, this open source revolution.
Mm-hmm.
We're going to be tearing into the technical details of these next-gen models. We're talking ESMFold, OmegaFold, RoseTTAFold, all-atom, or RFAA, and that really groundbreaking affinity predictor, Boltz-2.
Mm-hmm.
And we're going to compare them, rigorously against the established benchmarks, the AlphaFold family, looking at why and how they're succeeding, where maybe the pioneers stopped short.
And the sources we're drawing on here, they're highly technical. I mean, they're aimed squarely at a research AI audience like you listening. So we're really diving deep into things like scaling laws, these geometric architectures, how the models handle the complexities of small molecules, and the specific training strategies needed to unlock that binding affinity and even multiomics application potential.
Yeah, the core shift here feels really profound, doesn't it? The field is moving so fast. From accurately predicting just one protein chain to now natively predicting these entire complex biological assemblies, including metals, DNA, RNA, ligands, all together. And for the first time, getting close to the accuracy needed to actually predict binding function directly from sequence and context alone. I mean, that transformation is just redefining modern biology.
Okay, so to really understand these newer models, we probably have to start with the baseline, right? AlphaFold 2. I mean, it was revolutionary, no question. But its accuracy was fundamentally reliant on multiple sequence alignments, MSAs, and also templates.
Right, the MSAs.
Yeah, and the really time-consuming step in using AlphaFold 2, it's not actually the neural network calculation itself. It's the pretty labor-intensive process of searching these huge sequence databases like UniRef for all those evolutionarily related sequences just to build that optimal MSA.
And that MSA generation, that's the real computational bottleneck, isn't it? It's what stops it from being truly high throughput. I mean, if you want to predict, say, 1 million unknown protein structures from some metagenomic data, waiting potentially hours for database lookups for every single sequence, it's just prohibitive.
Exactly. Prohibitive is the right word. And that bottleneck is precisely what this first wave of next-gen models, especially ESMFold, was engineered to just smash through. And they did it by leveraging the power of these massive pre-trained language models.
Okay, ESMFold. So it uses the representations learned by this enormous ESM2 language model, right, to do accurate atomic-level structure prediction, but directly from a single protein sequence. It just bypasses that whole MSA search step.
Completely skips it.
Let's talk about the scale of that foundation model, ESM2, because that seems to dictate the performance here.
It really does. ESM2 was scaled up to, get this, 15 billion parameters. Now, for listeners familiar with large-scale deep learning, that's significant. It positions it as one of the largest protein language models evaluated to date. It's only about an order of magnitude smaller than major text LLMs like, say, GPT-3 at 175 billion, or some of Meta's Llama architectures.
Wow.
Yeah, and it's a scale that allows the model to effectively compress all that evolutionary and structural information into the latent space of just a single sequence representation.
And the immediate practical payoff of that scaling is speed. Just how dramatic is that efficiency? Are we talking small percentages or...
Oh, it's dramatic. The inference speed of ESMFold is an order of magnitude faster than AlphaFold 2. So for typical sequences, you're looking at maybe seconds, even milliseconds per prediction, compared to minutes or potentially hours for AlphaFold 2 if it's running a full MSA search.
An order of magnitude.
Yeah. And when you scale that up, think about predicting structures for sequences from these huge metagenomic databases. It suddenly becomes feasible within, say, days, rather than requiring months of heavy compute cluster time. It essentially transforms what was a really high-cost specialist endeavor into something more like a routine computational biology task.
Okay, but the obvious question whenever you take a shortcut like removing the MSA is accuracy. What's the trade-off? Did they lose a lot of precision?
The sources suggest the trade-off is surprisingly manageable, provided the sequence is, well understood by the language model itself. ESMFold achieves competitive accuracy for what they call low perplexity sequences. Basically, sequences where the language model shows high confidence and low uncertainty.
Makes sense.
So that 15 billion parameter model, for instance, achieved a highly respectable TM score of 71.3 on the Cameo test set. Now, where the language model struggles, or for truly novel sequences unlike anything it's seen, the performance gap between ESMFold and a full AlphaFold 2 prediction using a rich MSA does widen.
Okay.
However, and this is important, the sources really emphasize that when they artificially restricted AlphaFold 2 and RoseTTAFold to only use single sequence input, ESMFold decisively outperformed them. That really demonstrates the power locked inside that large language model's latent representation.
So ESMFold uses sheer scale and that deep language modeling understanding to win big on speed. Okay, let's pivot then to OmegaFold. It also achieves state-of-the-art single-sequence prediction, but its architectural focus seems quite different, shifting much more towards geometric principles.
That's right. OmegaFold takes an alternative, but you could argue equally powerful approach. It combines its own protein language model, which they call OmegaPLM, with this highly customized geometry-inspired transformer they dubbed the geoformer. The real breakthrough here is how it generates those high-resolution coordinates. It's not purely from evolutionary signals or just massive scale, but by actively enforcing structural logic.
The geoformer. The papers mentioned it's a deep stack, something like 50 layers. Now, given our audience knows the EvoFormer and AlphaFold 2 pretty well, what exactly is the geoformer doing differently inside? What's the core mathematical distinction?
Yeah, that is the core difference. While the EvoFormer and AlphaFold 2 is really designed to integrate information gleaned from the MSA, those evolutionary couplings, the geoformer, it focuses pretty much exclusively on enforcing geometric consistency. It iteratively processes the single sequence embeddings, basically ensuring that the derived amino acid node representations and the pairwise distance predictions translate into a physically plausible 3D space.
So it's acting like a deep geometric validator and refiner, almost. How does that kind of vector geometry focus translate into better predictions for targets where maybe that evolutionary data is sparse or missing?
Well, think about the underlying math, right? The geoformer uses principles derived from vector geometry to ensure structural invariance. Its layers are designed specifically to reconcile potentially conflicting distance and angular predictions that might arise just from the one-dimensional sequence input. This becomes a critical advantage for things like orphan proteins.
Ah, right.
These are proteins which have few or maybe even no related sequences in databases like UniRef. For those targets, AlphaFold 2's EvoFormer gets almost no signal to work with. But the geoformer can still enforce structural rules based on the intrinsic chemical and spatial constraints of the polypeptide chain itself.
And the sources specifically called out antibody prediction as a big win for OmegaFold. Why does that geometry-first approach work so well there? Antibodies aren't exactly orphans, are they?
No, they're not orphans, but they are challenging because they evolve so quickly. So while they often have many related sequences, the MSAs you get can be quite noisy. This is due to really high sequence divergence, especially in those critical functional regions, like the CDR loops.
Okay.
OmegaFold's geometric approach seems to cut through that noise. It relies less on those potentially misleading evolutionary signals and focuses more on local and global structural consistency, the result. Significantly lower RMSDs on those flexible but functionally critical regions, like the CDR-H3 loops. It really proves that for certain classes of proteins, maybe fast-evolving ones or sequence-sparse ones, geometric consistency might actually be a more reliable prediction signal than just evolutionary divergence. The success of these single-sequence models, it really forces us to look a bit deeper into the mathematical drivers behind them. The ESMFold research in particular gave us one of the most compelling insights into this relationship between language understanding and 3D structure. I'm talking about that inverse correlation with perplexity.
Right, perplexity or PPL. That's the standard LLM metric we use to quantify the model's uncertainty. Basically, it's confusion. Lower PPL means higher confidence in predicting the next token, the next amino acid in this case. A PPL of 1 means the model is perfectly certain.
Exactly. And the ESM2 research showed an almost perfect inverse correlation between that validation perplexity score and the final structure prediction accuracy. For the CASP14 TM score metric, the correlation was reported at -0.99.
Minus 0.99.
Yeah.
That's almost perfect inverse correlation.
It's incredibly strong. It's not just a high correlation. It really suggests a fundamental mechanism might be at play here. It sort of implies that structure prediction accuracy emerges emerges almost naturally as the language model simply achieves a deeper understanding of the language of protein sequences.
Wow.
It's like the 3D structure is somehow mathematically encoded within the high-dimensional latent space of that linear sequence, and just improving the language model's grasp of the sequence grammar directly unlocks the physical reality of the fold.
That is a genuinely staggering implication for foundation models in biology.
Yeah.
It suggests that maybe specialized structural training down the line might even become secondary to just pure scaling and achieving that deep linguistic comprehension of biological sequences.
It absolutely reinforces the power of scaling laws, yeah. And furthermore, their scaling analysis showed these highly relevant non-linear improvements. It's a phenomenon AI researchers will definitely recognize from general text LLMs. They saw these sudden, almost discontinuous jumps in RMSD improvement happening only at specific scale thresholds.
Like phase transitions.
Kind of, yeah. For instance, on a difficult CASP target, T1056, this was a novel, complex protein, the accuracy sort of plateaued until the model reached that 15 billion parameter scale. And then, bam, the structural prediction suddenly snapped into high accuracy.
Those emerging capabilities, right? It's a hallmark of scaling in text models, too, where a model suddenly learns to do complex reasoning or follow instructions, but only after hitting a critical parameter count. So here, the emerging capability is physically plausible 3D structure prediction.
Precisely. The T10056 example really demonstrates that for certain very challenging proteins, the level of complexity required to correctly model all those intricate internal contexts, the hydrophobic packing, it only resolves once the sequence representation gains sufficient dimensional depth from the scaling. Below that threshold, the model is essentially guessing. Above it, the structural logic just clicks into place. It's a powerful argument for continuing to push towards even larger protein foundation models.
Okay, so that's emergence through scale and perplexity. Now let's swing back to OmegaFold and that specialized geometry in the geoformer. If ESMFold uses scale to enforce emergence, the geoformer seems to use mathematical constraints to enforce consistency. Is that a fair way to put it?
That's a very elegant way to put it. Yes, that's the trade-off. The geoformer explicitly relies on fundamental geometric principles like the triangle inequality. You know, in basic Euclidean space, the sum of the lengths of any two sides of a triangle must exceed the length of the third side. If your predicted distance matrix violates this rule, your predicted 3D coordinates are inconsistent, physically impossible.
So the geoformer's layers aren't just applying standard attention mechanisms like in other transformers. They are mathematically optimizing the representations against this fundamental geometric axiom.
They absolutely are. The geoformer layers iteratively refine the predictions based on this. The sources highlight a really dramatic effect happening in the initial layers of the stack. Geometric inconsistency, which they measured by violations of that triangle inequality, it just drops precipitously early on.
How much?
In one example they cited, the inconsistency measurement plummeted from something like 0.638 down to 0.160 within just the first 20 layers of the geoformer stack. And concurrently, the contact prediction accuracy improved sharply. The remaining 30 or so layers then seemed to transition into more subtle refinement. It suggests that initial geometric stabilization is the most critical step, ensuring that abstract sequence representation gets correctly mapped to a plausible 3D topology first.
Okay, so we've established these two parallel pathways to accurate single-sequence prediction. Scale driving emergence via perplexity and geometry driving consistency via constraints. But what about ensemble learning? Is there still a role for just combining the pioneers, AlphaFold 2 and RoseTTAFold, especially now we have these powerful single-sequence models?
Oh, absolutely. The success of ensemble methods like RStack, which basically stacks AlphaFold 2 and RoseTTAFold predictions and feeds them into a meta-model, it really reaffirms a crucial truth. It's highly unlikely that any single method will be the optimal solution for every single protein out there, especially given the vast diversity of the protein universe.
How does RStack practically improve upon just running AlphaFold 2 or RoseTTAFold individually? What does the metamodel learn?
Well, ARStack essentially trains a neural network on the outputs of both of those baseline models. It learns the specific relationship between those two different predictions and the actual true conformation. And crucially, the sources found that the stacking approach was most beneficial in cases where AlphaFold 2 itself performed poorly, specifically on proteins where AlphaFold 2's RMSD was greater than about 1 angstrom.
Ah, so it learns to correct the errors.
Exactly. The ensemble learns to identify and potentially correct systematic errors or ambiguities that might have confused the individual models. It's like getting a second opinion and having an expert synthesize them.
And there was also a finding that stacking was particularly helpful for smaller proteins, right? Those under maybe 10 kilodaltons. Why would that be significant?
Yeah, that's interesting. Smaller proteins often have less distinct internal structure, fewer long-range contacts holding them together. They can be more flexible, maybe even a bit floppy. Their high-dimensional representations in the models can be more ambiguous as a result. So by combining two distinct structural predictions, the metamodel seems to gain enough redundancy or consensus to converge on the most accurate prediction. It effectively stabilizes the prediction for these more challenging, structurally fluid targets. It really makes the case that ensemble methods remain essential, even as the individual foundation models get more powerful.
Okay, so the models we've discussed so far, ESMFold, OmegaFold, they're really mastering the protein backbone, getting faster and more accurate. But biological function, I mean, it rarely happens in a vacuum, does it? It needs cofactors, metals, nucleic acids, small molecule ligands.
Absolutely.
Which brings us neatly to the next massive challenge. Generalization. And this seems to be addressed powerfully by RoseTTAFold all-atom, or RFAA.
Yeah, that generalization gap was, well, pretty severe in earlier models. If you can't accurately model the non-protein components, which include critical cofactors, modified residues, and importantly, drugs, you simply cannot model biological function accurately. RFA really tackles this head-on by moving from predicting coordinates just for the C-alpha backbone, maybe a few side chains, to modeling every single atom in a complex biological assembly all at the same time.
Every single atom.
Yeah.
That requires a huge shift in the model's architecture, its input vocabulary. How did RFAA actually handle this leap into full all-atom complexity?
Well, the input space just explodes, as you can imagine. RFAA is designed to model not just proteins, but also 8 different nucleic acid-based types, small molecules, metal ions, and even covalent modifications. To achieve this, they had to vastly expand the model's fundamental perception of what a sequence even is. So in addition to the standard 20 amino acids and the 8 bases, they introduced 46 entirely new element-type tokens just for non-polymer atoms like carbons, nitrogens, oxygens, and ligands or metal ions. It's effectively like adding 46 new letters to the alphabet of structural biology.
That's a massive expansion of the model's grammatical rules, so to speak. But how does it handle the specific chemistry of those small molecules? I mean, they're inherently complex atom bond graphs, not linear sequences like proteins. How does it integrate that alongside the protein sequence?
Yeah, that's a key technical innovation they introduced. The input features include explicit atom bond graph representations for the small molecules. These specify single, double, triple, or aromatic bonds, which are obviously critical for defining the chemical topology. These bond features get integrated into the 2D pairwise track of the model, alongside information about sequence proximity.
Sure.
And furthermore, RFAA incorporates stereochemistry. Specifically, it takes the chirality sign around each chiral center as input and integrates that into the 3D track.
Why is that stereochemistry input so critical? Why add that specific detail?
Well, chirality, the fact that many molecules exist as non-superimposable mirror images, is absolutely paramount in pharmacology. Drug binding is often highly sensitive to which stereoisomer you have. One might be active, the other inactive, or even toxic. By encoding the chirality sign explicitly, the model ensures structural invariance and physical plausibility. It prevents it from accidentally predicting a non-existent, or worse, the incorrect enantiomer, which is a major pitfall in traditional computational chemistry.
Makes sense. And optimizing this incredibly generalized system where you might have thousands of protein atoms, but maybe only 30 or 40 ligand atoms that must have required a specially tailored objective function, a loss function.
Exactly. They introduced an all-atom version of the frame-aligned point error loss, the FAPE loss that AlphaFold pioneered.
Okay, and why the customization? Standard FAPE loss, as I recall, focuses mainly on C-alpha distance errors in the protein backbone.
Yeah, the customization is vital because of that huge disparity in atom counts. Imagine you have a small ligand molecule, and maybe its 30 atoms are slightly misplaced. In a standard FAPE calculation, the error contribution from that tiny ligand might be completely dwarfed, just washed out, by the correctness signal from the 3,000 perfectly placed protein atoms.
Right, wouldn't even notice the ligand error.
Pretty much. So RFAA solved this very cleverly by upweighting the contributions to FAPE calculation that involve non-polymer atoms, ligands, metals, covalent modifications. This forces the model to achieve highly accurate, chemically plausible geometry for those small molecule components, too. It ensures sensitivity to critical details like bond lengths and angles in the ligand rather than just optimizing the overall bulk shape of the protein.
And this capability must have immediate, huge implications for drug discovery, particularly in things like blind docking, right? How did RFAA actually stack up against traditional methods when they validated it?
The performance was, frankly, a decisive validation of this all-atom approach. In blind Cameo ligand docking evaluations, Cameo is a continuous benchmarking platform, RFAA, consistently outperformed traditional non-deep learning methods like the widely used AutoDoc-Vina.
How much better?
On the set of cases where both servers actually produced a structure, RFAA successfully modeled 32% of those cases with a ligand RMSD under 2 angstroms, which is kind of the industry standard for a good prediction, a hit, compared to only 8% achieved by the Vina server. That's a 4-fold improvement. It's a dramatic jump in accuracy for blind docking, which could really transform the front end of virtual screening campaigns.
And presumably, its ability to handle complexity that traditional methods just cannot manage simultaneously. That's where the generalization really pays off, too.
Absolutely. RFAA is, as far as the sources indicate, uniquely capable of modeling complexes with multiple different non-protein components all in a single pass. The papers detail examples like, say, a DNA polymerase enzyme complex simultaneously with its bound DNA strand, an active site GTP molecule, the nucleotide, and a critical coordinating magnesium ion cofactor.
Oh.
Yeah. Previous methods would typically require sequential, multi-step docking and modeling for something like that, which often leads to cumulative errors stacking up. RFAA models the entire functional assembly, effectively a multi-omic system in one single unified prediction.
It also sounds like RFAA excels at modeling covalent modifications,
Yeah.
things like glycosylations, which are central to so many post-translational dynamics and are also critical drug targets themselves.
That's correct. Glycosylations, these complex sugar chains attached to proteins, are often poorly understood structurally, despite being crucial for things like protein stability, cell signaling, and immunology. RFAA achieved impressive accuracy here, predicting 46% of recent test cases under a 2.5 angstrom modification RMSD. It successfully modeled quite complex glycan chains, up to 7 monosaccharides long, even in target proteins that had low sequence homology to known structures. This capability is really pivotal for areas like vaccine design and understanding the mechanisms of things like antibody-drug conjugates, ADCs, where that covalent linkage is absolutely key to how they work.
Okay, so if RFAA can accurately model these incredibly complex existing structures, the logical next step is to flip the problem around. Can it design new ones? Tell us about RF Diffusion All-Atom.
Right. RF Diffusion All-Atom is essentially RFAA fine-tuned specifically for generative structural design using a diffusion model approach. So instead of just predicting a structure from a known sequence, it designs entirely novel proteins de novo from scratch. The process typically starts from a highly randomized input. They describe it as a Gaussian distribution of residue frames, and then it iteratively refines this noise into a stable, plausible, and hopefully functional protein structure, often built around a fixed non-protein motif that you specify.
That sounds like true rational molecular engineering.
Yeah.
Starting with a function or a binding partner and designing the protein around it. Was there solid evidence that these designs were actually functional experimentally?
Yes. The experimental validation reported was quite diverse and extremely strong. RF diffusion AA designs were proven in the lab to bind various small molecules, including the therapeutic compound digoxygenin. Critically, it successfully designed proteins that could scaffold the enzymatic cofactor heme, and these designs were highly thermostable, capable of maintaining their structure and function up to 90 degrees Celsius, which is remarkable. And perhaps most impressively, in a stunning demonstration of rational control over function, they designed novel protein scaffolds specifically to bind optically active bilin molecules. These AI-designed proteins achieved a significantly higher fluorescence quantum yield than previous scaffolds that had been painstakingly designed by human experts over years. This is powerful evidence that the model understands not just protein folding, but the intricate relationship between structure and advanced biophysical function, like light emission. Okay, so we've navigated speed with ESMFold and structural complexity with RFAA. Now we hit what many consider the final frontier in computational drug design: accurately predicting binding affinity. We're talking K, ED, IC50 values. This is the metric that truly separates a potential drug candidate from just an inactive compound.
And it's notoriously hard, right? Traditional docking methods are generally too imprecise for reliable affinity ranking, and the gold standard, things like free energy perturbation or FEP simulations, while accurate, are just far too slow and computationally expensive for large-scale screening.
Exactly. That's the critical gap the pharmaceutical industry has been trying to bridge for decades. Which brings us to Boltz-2. How did this model manage to enter the field and make claims of achieving FEP-level accuracy?
Yeah, what's the story there?
Well, Boltz-2 is reportedly the first AI model to truly approach the predictive accuracy of those gold standard FEP methods, at least on public benchmarks. For instance, the sources mention it achieving a Pearson correlation coefficient of 0.66 on a challenging 4-target FEP subset. That's getting remarkably close to the inherent noise level of the FEP method itself.
Okay, 0.66 is impressive correlation for affinity. But the efficiency claim is what really jumps out.
Right. The efficiency is what makes it potentially revolutionary. It's stated to be at least 1,000 times more computationally efficient than running those FEP simulations.
1,000 times?
Yes, 1,000-fold speed up. This could fundamentally change the economics and timelines of early-stage lead optimization basically overnight.
Okay, a 1,000x efficiency gain with comparable accuracy. That can't just come from building a bigger network, can it? It must involve some breakthrough in how they curated the data or designed the loss function.
It's definitely the strategy behind the training signal. Firstly, the team undertook a massive effort to standardize millions of inherently noisy biochemical assay measurements, applying rigorous filters to try and maximize data quality. But the real architectural breakthrough seems to be the customized loss function they use, which focus very strongly on pairwise interassay differences.
Okay, let's unpack that pairwise interassay differences. Why is focusing on the difference within a single assay experiment more informative for training than just using the raw affinity value itself?
It turns out to be the key to capturing these high-frequency variations in affinity, which medicinal chemists often call activity cliffs. You see, raw binding data from different labs, or even different experiments in the same lab, can have high interassay variance. The absolute numbers might differ significantly due to experimental conditions.
Right, comparing absolute IC50s across assays is tricky.
Exactly. However, the relative difference in affinity between two highly similar chemical compounds, when measured in the same assay under identical conditions, tends to be much more robust and reliable. By training the model specifically on these subtle, pairwise relative shifts, Boltz-2 learned to distinguish the functional importance of small structural changes, like the difference between a potent 10 nanomolar binder and a nearly inactive 1 micromolar non-binder that looked almost identical. That ability to predict those sharp activity cliffs is precisely what's needed for successful hit-to-lead optimization in drug discovery.
That makes perfect sense. It's basically training on the derivative, the change in affinity, rather than the absolute value.
Yeah.
Very clever. Beyond just predicting affinity, Boltz-2 also introduced some advanced controllability features that let researchers integrate their prior knowledge more seamlessly.
Yes, and this is critical for making these models practical in real-world discovery pipelines. Boltz-2 can actually be conditioned on the type of structural input being used, whether it's derived from X-ray crystallography data, NMR ensembles which show flexibility, or even trajectories from molecular dynamics, MD simulations.
So you can tailor the prediction to the context.
Exactly. It allows users to test specific hypotheses or align the predictions to the particular conformational ensemble they expect under different experimental conditions. For instance, if you suspect a target protein is highly flexible, you could condition the dynamic-aware affinity estimate.
And that integration of dynamics seems really fundamental to Boltz-2's design, doesn't it? It didn't just rely on static PDB structures.
That's absolutely right. Local protein dynamics, things like induced fit upon ligand binding, are often essential for understanding affinity accurately. Boltz-2 was explicitly trained using curated molecular dynamics trajectories from large datasets like Masato and Atlas. It was also supervised directly on B factors, which measure atomic motion extracted from both experimental structures and computational simulations.
So it learned dynamics.
It learned to incorporate dynamics. This seems to be the first time a structure and affinity predictor has successfully integrated dynamic information so deeply into its training. And apparently, conditioning the model on MD inputs during inference leads to predicted structural ensembles that show stronger correlations with experimentally observed local protein flexibility measured by RMSF compared to purely static models.
And on the architectural side, there was also mention of an improvement in how it uses template information, using something called physics-based steering potentials.
Yes, this really elevates the hypothesis testing capabilities. Firstly, Boltz-2 natively supports multimeric templates, meaning you can provide structural information about protein complexes, not just single chains. But the steering potentials allow for the strict enforcement of user-defined constraints during the prediction process.
Like forcing certain contacts.
Exactly. For example, if a researcher has experimental data, maybe from cross-linking or cryo-EM, suggesting that two specific residues must be in contact in the bound state, they can apply a physics-based potential that essentially steers the model's diffusion process to strongly respect that distance constraint. This helps ensure the output structure is not just statistically probable based on the training data, but also physically and experimentally plausible according to their specific prior knowledge. That's vital in iterative lead optimization campaigns.
This whole framework, accurate affinity, controllability, dynamics, constraints, it really strongly suggests an end-to-end pathway for actually finding new, verifiable drug molecules, doesn't it?
That definitely seems to be the realized goal, yes. The sources detail how Boltz-2 is coupled with another model called SynFlowNet, which is a specialized generative model focused on creating synthesizable chemical compounds. They use this combined pipeline to perform large-scale virtual screening.
And did they validate it?
They did. They validated this generative pipeline prospectively against a real drug target, the TYK2 kinase. They identified high-affinity candidate molecules computationally, and then crucially, they rigorously confirmed these predictions using absolute FEP simulations, which they actually implemented within their framework and dubbed Boltz-ABFE. It demonstrates a complete high-confidence cycle, going from sequence to structure, predicting affinity, generating novel and synthesizable designs, and then getting high-confidence validation, all driven by these advanced AI tools.
And finally, the reach of this Boltz-2 model seems to extend far beyond just small molecule drug targets through what they call its multi-omics extension.
Indeed, Boltz-2 significantly augmented its training data set by using distillation, basically, learning from the predictions of an earlier version. Boltz won across a range of complex biological interaction modalities. This included things like RNA interactions, protein-DNA interactions, RNA-ligand complexes, and, critically for immunology, interactions between T-cell receptors and peptide MHC complexes, TCR-PMHSC.
So it learned patterns from all these different interaction types.
Exactly. By distilling knowledge across these diverse multi-omic predictions, Boltz-2 is potentially positioned to apply its core affinity prediction methodology to fields far beyond traditional small molecule drugs, like immunology, diagnostics, and genetics. This could be absolutely essential for developing more sophisticated disease risk prediction models in the future, which will need to accurately model complex interactions within vast and diverse biological networks. It really extends its utility towards things like biomarker development and understanding functional genetics. Okay, let's maybe pull back for a moment and look at the sheer scale of practical impact that's already being realized, specifically thinking back to ESMFold. That speed advantage we talked about, it's not just some academic metric, it's proving to be a powerful tool for exploring what you might call biology's dark matter.
Ah, you mean the metagenomic exploration aspect. Using that order of magnitude speed gain to actually map the vast unknown protein universe hidden in environmental sequencing data.
Precisely. ESMFold's speed made it practical, for the first time really, to rapidly compute something like 1 million predicted structures directly from sequences in the massive MGenify metagenomic sequence database. And this was done within practical timescales, weeks or months, not years. This represents a huge chunk of the protein universe that was previously structurally invisible.
And the findings, when they looked at that new structural space, were they significant? Did the model reveal much genuine structural novelty?
Profound novelty, according to the reports. They found that a large fraction of the high-confidence structure predictions, the ones the model was really sure about, had very low structural similarity, measured by TM score, often less than 0.5, to any structure currently deposited in the known protein databank, the PDB.
Wow.
And furthermore, many of these novel structures also had low sequence similarity to entries in sequence databases like UNIRF90. So this means they are likely fundamentally new sequences and new folds, yet the model was still highly confident in predicting their 3D structure.
So ESMFold is essentially providing structural insight where the sequence data alone gave you almost nothing to go on, enabling structure-based functional assignment for these unknown proteins.
Exactly that. The sources cite specific cases where an mGNFI sequence had no meaningful sequence match in UniRef90, making traditional functional annotation basically impossible, yet its ESMFold predicted structure aligned beautifully with known functional classes, like, say, specific types of nucleases or known lipid binding domains. This allows for immediate structure-based functional hypothesis generation for these completely novel genomic elements discovered in metagenomes. This kind of application is really foundational for things like discovering new enzymes, biomarker development, and just fundamentally understanding environmental or microbial biology at a deeper level.
Okay, bringing this all together then, we've witnessed these almost simultaneous revolutions happening. Incredible speed in emergent structure prediction through LLM scaling with ESMFold, generalization across pretty much all chemical elements and complexes with RFAA, and now high accuracy, high efficiency functional prediction, especially affinity, with Boltz-2. Given this somewhat fragmented landscape of specialized successes, what does the research seem to suggest about the inevitable future of structural biology AI? Is a one model going to rule them all?
You know, the convergence of evidence, especially when you look at the analysis of ensemble methods like Aerostack that we discussed, it really leads to a pretty clear conclusion. It seems highly improbable that any single unified method will ever outperform all others across the entire immense diversity of the protein space.
So specialization is key.
It looks that way. The future almost certainly lies in ensemble prediction and in highly specialized foundation models. We're likely to see, and are already seeing, foundation models explicitly designed and trained for specific tasks, maybe RFAA and its successors for multi-component complexes, Boltz-2 and similar models for precise affinity optimization, perhaps OmegaFold-like architectures for orphan proteins or antibodies. The real power will come from integrating these specialized tools into comprehensive, highly automated prediction pipelines. This deep dive really confirms that the field seems to be moving decisively away from the single model dominance we saw initially with AlphaFold 2 toward a much more diversified, vibrant, and importantly, open source architectural ecosystem.
So wrapping up, what does this all really mean for us, for the field? We've seen the journey, haven't we? Moving from that monumental single-focused challenge of just predicting one protein chain accurately to now tackling speed, geometric consistency, modeling all atoms and complex assemblies, and even hitting that holy grail of drug discovery, predicting binding affinity with something approaching FEP-level accuracy. And so much of this driven by open-source innovation. It feels like an acceleration beyond anything we've really seen before in computational biology.
Absolutely. And maybe consider this for your own work, for how you think about these tools. Boltz-2 has shown that it might be possible to achieve the accuracy of free energy perturbation, which is often the metric needed for really confident decision-making, sometimes even required by regulators, but potentially 1,000 times faster. Now, is integrating these high-accuracy, high-speed affinity models with generative chemistry pipelines, like SynFlowNet, genuinely allows drug discovery teams to rationally design, prioritize, and then experimentally validate novel synthesizable binders in, say, days or weeks, instead of the typical multi-year cycle. Just think, how will this completely redefine the economics, the timelines, the very possibilities for tackling historically intractable disease targets? The true revolution, perhaps, isn't just the prediction itself. It's the speed and fidelity of rational molecular engineering that we've now unlocked. That's where the really exciting work begins.